Insulin was the first hormone to be ever discovered in the year 1921. And it was discovered by two scientists, that is Charles Banting and Frederick Best. Okay, so these scientists, what did they do? They did experiments on a dog whose pancreas was removed, and they had to find out that the principal hormone that controlled the blood glucose levels was insulin, whereby they injected the insulin to the dog progressively and it had to improve. So that's not what our interest is now. Our interest is to study what insulin is, the structure, the release, and the mechanism of action. So let's begin. So this is the 3D structure of insulin. This 3D structure of insulin was determined by a very important scientist called Dorothy Hodgkin. Okay, so Dorothy Hodgkin is one we give up, is a scientist we must give applause to. Okay, so Dorothy Hodgkin. So what did she do? She used a technique called X-ray crystallography to determine the 3D structure of insulin. Okay, the insulin that we now know and we can manipulate to produce different types that we use in treating diabetes. Okay, so X-ray crystallography is that procedure that was used by that scientist to determine the 3D structure of insulin as we see. Okay, and in the determination, what does the structure of insulin really have? It has 3 disulfide bonds, as you can see. There are 3. Then what else does it have? You know, insulin has a molecular weight of 5,800 and has what we call an isoelectric point of 5.4. So what does isoelectric point mean? Isoelectric point is that pH, okay, at which a molecule is neutral. In other words, it doesn't have a positive or a negative charge. It is completely neutral. And for that isoelectric point for insulin, where it is completely neutral, it is 5.4 arbitrary units. Okay, so that's what we should know. Then, from that technique still, you find that the structure of insulin, besides having these 3 disulfide bonds, okay, it has 2 polypeptide chains. The first polypeptide chain is what they call polypeptide chain A, which is this. And the second one is what they call the polypeptide chain B. The polypeptide chain A has 21 amino acids, and the polypeptide chain B has 31 and has 30 amino acids, making the whole total to be 51 amino acids. 21, 30. Point taken home. So, where does this insulin come from? Insulin basically is produced by the pancreas, okay? You know, this is the pancreas. It is a very important organ in the body that is responsible for production of hormones. It plays a role in both exocrine and endocrine function. So you find that within the pancreas, we have cells, the islets of Langerhans, okay? So if we look at an islet of Langerhans, well, it has basically 2 cells that we should know that are helpful in controlling the amount of glucose. So basically for our focus here, we shall focus on insulin, though insulin that is produced by the beta cells, okay, of the islet of Langerhans. The beta cells, they're the ones that produce insulin. But we also have other cells in the pancreas that produce what? Different hormones. For example, we have for glucagon, we have the alpha cells that produce glucagon. We have the delta cells that produce the inhibitory hormone, somatostatin, okay, and amongst others, pancreatic polypeptide and so on. So, these beta cells, they are the ones that are responsible for production of insulin, that hormone we are talking about. That's the point that we should know first. Then, if you look at this, this is a structure of a beta cell. So, if we extract the beta cell, this is how it will be. But before doing that, I want us to first focus on an important concept. That concept of the blood glucose level. You know, the normal blood glucose concentration in your body is 70 to 100 milligrams, okay, per deciliter of blood. So you find that if you take a drink, this will increase, okay? But what we have in the hospital is normally the glucometer. What does the glucometer measure? It measures the blood glucose levels, the fasting blood glucose levels, but in millimoles per liter, okay? So this is the same as 3.8 to 5.6 millimoles per liter. We shall look at it more when we are discussing diabetes, okay? So this is the norm. You know, this is the normal blood glucose concentration, the fasting blood glucose level. fasting blood glucose sugar, what we can call the FBS. Okay, so what point am I trying to bring out here is that this is the normal amount of glucose, in that if you go past 100, you're in a state of hyperglycemia. If you're less than 70, you're in a state of hypoglycemia. Some books will tell you it is 80 to 120, but principally, traditionally, it is this. Okay? So, with this, what else must you know? When you go above the 100, that is when insulin will come to be released. For example, if you maybe take a cup of tea, you take maybe a bottle of soda, or even eat a slice of bread, you find that the blood glucose level increases. When the blood glucose level increases, the body must produce insulin. Insulin is that hormone that I've told you about, discovered in 1921, that will decrease the blood glucose levels back to the norm. Okay? If it is persistently high, if the blood glucose are persistently high, it means you have diabetes. Okay? So, it is so crucial for us to understand that. Leaving that alone, you find that when the blood glucose levels are high, how does the body try to reduce them? There is what we call reduction in absence of insulin, okay? Because of the receptors, okay? The receptors or even the channels. Receptors and channels. So I'm going to explain here. So imagine this is one beta cell, but one structure of a beta cell is this, okay? So let's take this for a beta cell. So this is a beta cell. So if the blood glucose level is high, okay, it goes above 100 milligrams per deciliter. The body must bring it back to the set point. So what happens? I'm going to use G for glucose, okay? So what happens? The blood glucose level in your blood now has increased. We have these channels, what we call the glucose transporter 2 channels. The glucose transporter 2 channels are very helpful in regulating the amount of glucose, okay? So this is the GLUT2, the glucose transporter 2. Though, on the beta cell, we have the glucose transporter 2. There is an argument that has been ongoing, or a confusion that has been ongoing, between the glucose transporter 2 channels in the humans and in the rodents. And people were saying, or the scientists were saying, some of them were saying that the glucose transporter 2 is absent in the human being. No, it is present in the human beings, okay? And it is present in the human being, though in a less concentration, or in a lesser concentration compared to that in the rodents, okay? So it is very important to know that human beings have the glucose transporter 2 channels that are glucose independent, okay? So if we look at briefly about the glucose transporter channels, we have glucose transporter 1, we have GLUT1, we have 2, 3, we have 5, okay? These ones, they are glucose independent. They do not need, they're insulin independent. They're always working, even in the absence of insulin, okay? This one, glucose transport channel 1, is always found in the blood-brain barrier, okay? Allowing for exchange of blood into the brain CSF, okay? Then the glucose transport channel 2 is present in the liver, in the pancreatic B cell, okay? Then the glucose transport channel 3 is always present in the neurons, allowing for entry of glucose into the neurons. Glucose transporter 5 is always also present in the spermatocytes, allowing for the entry of glucose into the sperms, okay? Aiding its movement and doing other activities. Then we have the glucose transport channel 4. That one is specific. It is insulin dependent, okay? You need to know that because they can trick you on that, okay? Then other channels are also there, like the sodium glucose transport channel 1 and 2, that we shall see certain drugs, okay? Yeah, that work on that with diabetes. So it is important for us to continue. So if we focus on the glucose transport channel 2 that is present on the beta cell, why are we focusing on this? It's because the glucose transport channel 2 is bidirectional, in that it is pressure. It allows entry and exit of glucose into the cell. But how does that happen? For it to be maintained inside, it depends on a hormone called, and on an enzyme called hexokinase that we looked at in glycolysis, okay? So with that, I needed to recap that very fast for you. So when glucose enters into the cell, okay, it will undergo glycolysis. It undergoes glycolysis. It undergoes the Krebs cycle. It undergoes the Krebs cycle. From the Krebs cycle, it will undergo the electron transfer chain in the mitochondria. Then this will form ATP, adenosine triphosphate. Okay? Adenosine triphosphate. Adenosine triphosphate is the energy that your body recognizes and uses in that form. Okay? Basically, that is it. Now, leaving that alone, on the other side of the membrane still, of the cell membrane, we have what we call the potassium ion channels. The potassium ion channels are so important in the production or actually in the secretion of insulin. How? Normally, this channel, what happens with the potassium ion channel? Okay, this is the potassium ion channel. Let me write it here. Potassium ion channel. So this potassium ion channel, it allows for exit. Normally, it allows for exit of potassium ions into the extracellular space. Okay? Why? Because inside the cell, there is a lot of potassium compared to the outside. Because of that, the potassium ions that are inside the cell will leave into the outside part of the cell. So what will that cause on this? Okay, what does it cause on this? You find that because of the exit of the potassium ions out of the cell, you find that the inside of the cell is normally negative. So the voltage, the membrane voltage that is there is towards the negative bit. Okay, the millivolts, they are not strong enough. They are not positive. So where have I brought in this? I've brought it to explain to you the relationship between the ATP and this channel that will lead to the release of insulin. This is how it happens. So when the glucose concentration is high, okay, this concentration, when it is high, it will translate to this. Glucose enters, undergoes that to form ATP. So when ATP is too high, what happens? ATP being very high, okay? This increase in the ATP, or even what they call an increased ATP to ADP ratio, it will result into the closure of this channel, okay? The channel will close. So when the potassium ion channel closes, what happens? The closure of the potassium ion channel will increase the intracellular concentration of potassium ions. When the intracellular concentration of potassium ions increases, it will generate now a positive voltage, okay? A positive membrane voltage, basically up to around 40, up around to 30 or 40 millivolts, okay? The 30 or the 40 millivolts, they are strong enough to cause opening of the calcium ion channels, okay? So when this positive membrane voltage is generated, it will be transmitted along the cell membrane or the plasma membrane of the beta cell till it reaches the calcium ion channels. So at the calcium ion channels, what happens? You find that there is an entry of the calcium ions. How will it happen? You know, at the outside, there is a lot of calcium compared to the inside, okay? So when that stimulation happens of this channel because of the action potential or the membrane voltage that has been generated, you'll find that there is an increase in the intracellular concentration of the calcium ions, okay? And no calcium ion is necessary for the release or the exocytosis of the insulin, okay? Releasing it in the bloodstream so that it goes to the different target organs and it exerts the effect, reducing the blood glucose concentration, decreasing gluconeogenesis, increasing lipogenesis, right? Very, very important. So when calcium enters, what happens? Always inside the cell, there is what they call the insulin vesicles or the insulin containing vesicles. So these insulin containing vesicles, they are inside of the cell like that, and they contain insulin, okay? You know, insulin, there are very many things alongside the production of insulin. It comes along with different things. For example, the C-peptide that we shall use even in the diagnosis of diabetes. So it is very, very important. So inside this vesicle, there are important molecules in that. First of all, there is the C-peptide. C-peptide is very useful in determining what type of diabetes someone has, type 1 and type 2. Remember, with type 1, we say it is autoimmune destruction of the beta cells. It means they lack what? Insulin. How do we differentiate? It means in type 1 diabetes, we shall not see the C-peptide. But in type 2, we shall see it. That's how we can distinguish if someone has type 1 or type 2. Let's leave that for now. We shall do it later. Then we have the insulin itself. Okay? Though these two come together, we have the pre-pro insulin, then we have the pro-insulin. Okay? So, the pro-insulin contains this. Okay? Then we can have what we call the amyloid beta protein. Okay? Is also another common one. The amyloid beta protein, and we shall see how helpful it is. Okay? Amyloid beta protein. And there's a certain enzyme that the scientists in Mayo Clinic discovered in the USA. I'll tell you about it. But for now, let's first continue. So, when we have this vesicle that contains all this, okay, because of the entry of the calcium ions, these calcium ions will act to allow the movement of different molecular proteins such that they cause the fusion of this vesicle with the cell membrane, thus releasing the insulin out. Okay? So how will it happen? You find that there will be fusion. Let me pick out this part like this. There will be fusion of this vesicle with the cell membrane, okay, in such a manner. Okay? When it happens in such a manner, you'll find now with that, we shall be able to release our insulin. Okay? So, by exocytosis. Okay? This is now the insulin. This insulin is already now into the bloodstream. Okay? That. The insulin goes into the bloodstream. The vesicle fuses with the plasma membrane. The insulin goes into the blood circulation. Just like that. So, how do we continue from this? You find that when we have the insulin now in blood, the target organ first is the liver. It will first go to the liver to meet a very, very important enzyme, a zinc metalloproteinase enzyme or a zinc metallopeptidase enzyme that I'm going to talk about. We call it the insulin degrading enzyme, IDE. Right? So, let's talk about it here. So, with this, first of all, it will go to the liver. Okay? The liver is the first thing that this insulin will go to. So, if we have the liver around like this, right? You find that this insulin will first go to the liver. Okay? So, while in the liver, there is an important enzyme. That enzyme is called the insulin degrading enzyme, IDE. So, that insulin degrading enzyme is a very, very important enzyme, okay, that is helpful in degrading insulin as well as the amyloid beta protein, okay? So, you find that with this enzyme, it has very important hydroxamates that help it to work, okay? So, the different hydroxamates enable the decrease or the degradation in the amount of insulin before it goes into the whole circulation, okay? Why does it happen like that? The reason as to why it happens like that is that this insulin, about 50 to 60% of it must first be degraded in a way that it undergoes what they call the first pass effect. What we can look at in also in some drugs. Some drugs that are taken orally undergo what they call the first pass effect. So even with insulin will undergo the first pass effect or first pass metabolism in the liver, whereby 50 to 60% of it, okay, is degraded or used up by the liver. 50 to 60% of it is used up, and the rest sent into the systemic or the peripheral circulation. This one is via the portal vein. from the portal vein, then into the systemic circulation. So after that, the rest of the insulin will now be released into the body. Okay, the remainder of this. So this one will now go into the circulation. Okay, basically, what the transition between this step to this is the most important thing that you should know is this. Okay, the insulin degrading enzyme, which is a zinc metalloproteinase or the proteinase enzyme. Okay, so when you have this insulin now, maybe when you subtract 50 to 40%, when you've now remained with 50 to 40%, this insulin will go to the different organs. So those different organs, how you can recall them, you just know what the mnemonic LAMB. Okay, LAMB. In a way that the target organs first for insulin. This mnemonic shows you the different target organs for the insulin. The L stands for the liver. Okay, the A stands for the adipose tissue, right? Very, very important. The M stands for the muscle cell or the musculoskeletal cell, the muscles. Then the B stands for the brain. Okay, but basically, now this is where the other effects come in. But basically with the brain, we have the glucose transport channel 1 that I talked about. Okay, so our main focus now will be about the adipose and the muscles. Because the adipose and the muscles, now this is where the other thing comes in. With these ones have the glucose transport channel 4. Okay, the glucose transport channel 4. The glucose transport channel 4. This one is insulin dependent. It depends on insulin to take in glucose. Okay, so with that, we shall be able to start the mechanism of action of insulin. So now, when insulin reaches these organs, for example, imagine this one. Imagine this cell is a muscle cell or it is an adipocyte. Okay, it is not necessarily the real thing, but for you to understand the concept, because if you look at the histology of the adipose tissue, it has the white and the yellow fat, okay, that have different arrangements with the nuclei peripherally displaced. We shall look at that. Okay, so for this, let's take, maybe we use the same illustration so that we understand the basic concept of how insulin works. So with this part of the video, we shall be understanding the mechanism of action of insulin. So how does insulin work at this moment? You find that at this time now, this receptor is what, first of all, this is the receptor. Okay, that receptor is what they call the receptor tyrosine kinase. What is receptor tyrosine kinase? It is the insulin receptor. A receptor is like, in enzymatology, we talk about the mechanisms, okay, of enzyme activity, whereby we have the induced fit or even the lock and key hypothesis. So this will be like a lock and key hypothesis that triggers different intracellular reactions within that specific cell. Okay, so to continue with that, you find that when first of all, this insulin receptor, when we are talking about the insulin receptor is what we call a tetramer. Okay, it is a tetramer. It is a tetrameric protein. When they say tetramer, what does it mean? It means it has 4 main subunits. As you can see, 1, 2, 3, 4. There are 4. No discussion. So that's why we call it a tetramer, made up of tetrameric proteins. Okay, so in being a tetramer, the outer units is what they call the alpha. These two, it has the alpha 1 and 2. Okay, this is the first one, this is the second one. Then that's domain on the extracellular part or the extracellular domain. Then in the intracellular domain, okay, or the transmembrane domain, it has the beta subunits. Okay, so basically, quick point, it has 2 alpha units and 2 beta units. The 2 alpha units of the insulin receptor chain, of the insulin receptor, okay, the 2 alpha are extracellular, then the transmembrane and the intracellular, there is the 2 beta. There are 2. So what happens? Basically, we call this a tyrosine kinase receptor for insulin. But what is tyrosine? You know, tyrosine is a very, very important amino acid. If we are to look at this structure, tyrosine, you know, tyrosine, it has a structure like this. A benzene ring, right? That's the benzene ring it has. Then it has different subunits of it. It has here the oxygen atom, the hydroxyl group, right? It has the amino group there. Then on the other side, it has the hydroxyl group. That is what we call tyrosine, okay? It is a very, very important amino acid. So you find that this receptor has residues of tyrosine, okay? So this insulin receptor has residues of tyrosine that we can put on this other side. TY, TY, okay? Very, very important. So what happens? Now we are starting the mechanism of action of insulin. When this insulin comes, okay, when the insulin is released from the liver, it will move, okay, in the bloodstream, then it will bind to its receptor. When insulin binds to its receptor, it will trigger a certain series of reactions that are known basically as autophosphorylation mechanisms. Though when it enters deeper into the cell, it undergoes different series of phosphorylation and dephosphorylation, where there is simultaneous removal and addition of phosphate groups. Okay? Don't worry. So, when this insulin now binds to its receptor, okay, the receptor tyrosine kinase, what happens? You find that because of the binding to the alpha subunit, a reaction called dimerization will occur, which will lead to change in conformation. Okay? What does it mean? When insulin binds, okay, dimerization will occur, causing the two alpha units to come close together, changing the conformation, which will later affect the conformation of the beta subunits. So when the conformation of the beta subunits that contain the tyrosine residues occurs, autophosphorylation happens immediately. Okay? So autophosphorylation, addition of phosphate groups automatically. Okay? These phosphate groups are necessary to act as supplementation or even stimulatory effects to the proteins, which proteins we call the insulin receptor substrate proteins. Okay? So those insulin receptor substrate proteins, we have insulin receptor substrate protein 1 and insulin receptor substrate protein 2. Insulin receptor substrate protein. Okay? So there will be interaction between the phosphorylated tyrosine residues and the insulin receptor substrates. So with that, still autophosphorylation occurs, whereby you will be adding a phosphate on different sides of the insulin receptor substrate protein. Okay? So this insulin receptor substrate protein, it will, if it is phosphorylated, it is activated to act as a docking site. Okay? A docking site is like an attachment site. Okay? So what does this docking site do? This docking site enables attachment of an important molecule, what we call PI3K. Okay? PI3K, this PI3K stands for phosphoinositide 3 kinase. Okay? Phosphoinositide 3 kinase. So this phosphoinositide 3 kinase, it is a very, very important protein that helps in the transition of what they call the phosphoinositide to diphosphate to phosphoinositide triphosphate. Okay? How? It exerts its stimulatory effect, okay, in such a manner that it will catalyze the conversion of membrane lipids or membrane substrate. Okay? Phosphoinositide diphosphate or phosphatidylinosyl diphosphate to phosphoinositide triphosphate. Okay? It will stimulate that. So you'll have a conversion of that to that. So this PIP3 is very, very important in a way that it stimulates an enzyme that also will catalyze further reactions. Which enzyme will that be? The enzyme will be what they call the phosphoinositide dependent kinase. Phosphoinositide dependent kinase. Okay? Or phosphoinositide dependent kinase, 1, PDK1, okay? When that stimulation happens, this enzyme will be activated. What is the importance of activating this enzyme? When you activate this enzyme, it will also cause activation of another enzyme, okay, in a cascading effect, which enzyme we call protein kinase B, okay? When you stimulate protein kinase B, it will also go and increase the activity of the AKT, okay? AKT. So what is AKT? AKT actually, it is almost like a gene, or a gene, a gene coding sequence that was discovered in a retrovirus, okay? That is helpful also in such a process. So what does AKT do? AKT does very, very important effects, okay? So the most important effect in regulating the amount of blood glucose is the insertion of these channels. So this channel is what they call the glucose transport channel 4, okay, which is present in the muscle cells and also in the different other cells, okay? But basically the adipocytes and the muscle cells, okay? So how does that happen? You know, what does AKT do? The first thing AKT does, okay, to reduce the amount of glucose, it will cause, okay, through transcription mechanisms, okay, it causes the formation of vesicles that contain the glucose transport channel, the glucose transporters, okay, as this. So imagine you have it like this, okay? So what will happen? Because of the AKT, you'll find that there is fusion or insertion of these glucose transport channels into the cell membrane, resulting into this, okay? Now, when you insert the glucose transport channels, what happens? Still, this glucose will insert itself, or glucose will enter into the cells, okay? So this will cause entry of glucose into the cell, okay? It will cause glucose entry into the cell. So you'll have glucose into the cell. So when glucose is inside the cell, how does it relate to this? Remember, we had it was 70 to 100 milligrams normal, but it had increased above 100 milligrams per deciliter. So with the entry, you'll find that this entry will cause us to come back to the norm, okay? It returns to normal, right? That is how insulin works, okay? First of all, in a recap, how does insulin work? Insulin binds to its tyrosine receptor kinase receptor, triggering dimerization of the alpha subunits of the receptor, resulting into a conformational change, which will stimulate the beta subunits, okay? When the beta subunits are stimulated, autophosphorylation of the tyrosine residues occurs. Because of autophosphorylation of the tyrosine residues, it will give like a regulatory activation to the insulin receptor substrates 1 and 2, okay? Because of that, it will autophosphorylate them, resulting into activation, which acts as a docking site for PI3K. PI3K converts PIP2 to PIP3. PIP3 activates PDK1. PDK1 activates PKB. PKB activates AKT. AKT causes gene transformation, okay? So this is a gene expression rather, okay? Which results in formation of the vesicles which have the glucose transport channel 4, okay? Then allows for entry. Then also, there is a parallel pathway, okay? There is what they call a parallel pathway, okay? Or a parallel alternative. Like from the AKT, you will not go this side, okay? But basically, first of all, if I'm complete what AKT does. So with AKT, what will it do? You'll find that because of this, there is decreased blood glucose, okay? Resulting into a decreased blood glucose. 2, it will increase lipogenesis, okay? Lipogenesis, okay? 3, it will decrease gluconeogenesis. Gluconeogenesis. So these are some of the ones that it does, okay? So with that also there is an increase, okay, in glycogenesis, or the formation of glycogen. But how do this happen, okay? Specifically with gluconeogenesis, there is an important transcription factor, okay? So that important transcription factor is what we call the FOXO, okay? FOXO1. FOXO stands for the fork head box 01 protein, or transcription factor rather, okay? That is helpful in controlling the amount of gluconeogenesis. Then also the inhibition, okay, of what they call the glycogen synthase 3 kinase. So when you inhibit this, that is when you'll be able to increase glycogenesis. So all these happen because of the AKT. So this is a very, very important molecule, the AKT, very, very important, that allows for insulin to exert its effects, okay, as such. Then we have parallel pathways, okay? So with the parallel pathways, as I earlier on tried telling you, so with the parallel pathways, there are basically two, okay? But these ones basically they'll lead to, protein synthesis, cellular growth, gene expression in all ways. So basically with the parallel pathway, there's what we call, the parallel pathway comes off exactly from the AKT. So with that, you'll find that we have the mammalian target of rapamycin, the mTOR, okay? What we can call mammalian target of rapamycin, okay? Mammalian target of rapamycin, So with this one, when it is stimulated with that, what happens? You find that there is an increased production of cells, okay? Like what they call cell division, cell growth, or even gene expression to form different other proteins, or to act through different mechanisms that aim at decreasing this amino acid synthesis. All that happens because of that. Then the last one is what they like abbreviating as the MAPK pathway, okay? So MAP, this one basically it is for, mitosis, like all this stands for mitogen activated protein kinase, okay? So with this one, it will result into cell division, cell growth, and so on. But how does it do that? There are different stimulatory mechanisms, okay? Through the RAS and RAF MEK pathway, okay? So with this, you find that this one, it is basically for gene expression, which enables in cellular increase of mass or even other forms of gene expression. Though it is not so major, but the major ones are these, okay? But it's important to know that. So that's the mechanism of action of insulin, okay? Basically, that's it. Thank you very much. Keep on subscribing to the channel. Amen.